In today's lesson, we will be covering the Golden Gate assembly method. The Golden Gate assembly is a method to assemble fragments of DNA into a larger construct, similar to how we put puzzle pieces together. Fun fact: the name Golden Gate assembly came from the Golden Gate Bridge. Just like how the Golden Gate Bridge connects two land masses, the Golden Gate assembly connects separate DNA fragments together. The advantages of using the Golden Gate cloning method include high efficiency, seamlessness, high accuracy, and its many applications. To begin the Golden Gate assembly, it is first necessary to determine which specific DNA sequences you would like to combine into one cell so the cell can express a specific characteristic. Once that is determined, special enzymes called type 2S restriction enzymes like B-cell or BSMBI recognize specific DNA sequences and cut the DNA at a location close to the sequence. This makes Golden Gate assembly able to cleave many different sequences of DNA. When these enzymes cut the DNA, it creates overhangs or sticky ends with unique sequences on each fragment. Then, the DNA fragments with complementary overhangs are mixed together in a single tube. The overhangs of these fragments are different and can hybridize or stick together because they are complementary to each other. To permanently join these fragments, a DNA ligase enzyme and ATP are added to the tube. The DNA ligase acts like molecular glue, connecting the fragments by sealing the gaps between them. The resulting DNA construct can be transformed into bacteria or other cells, which allows DNA to be replicated and used for various applications. The Golden Gate assembly method is a useful tool for molecular cloning that accurately and efficiently assembles multiple DNA fragments with specific sequences into a larger DNA sequence. It uses type 2S restriction enzymes such as B-cell or BSMBI, which recognize specific DNA sequences but cleave the DNA at a defined distance away from their recognition sites. After GGA is used, successful recombinants can be screened for. Thanks for watching!